pleasant day to everyone who is watching. Today, we are ecstatic to delve into a revolutionary realm where technology meets healthcare: the application of recombinant DNA technology in diagnostics. In recent years, the landscape of healthcare has witnessed an extraordinary transformation propelled by advancements in biotechnology. One of the key players in this paradigm shift is rDNA technology, a groundbreaking innovation that has not only unlocked the secrets of our genetic code, but has also paved the way for a new era in diagnostics. So before we explore its application in diagnostics, let's briefly understand what rDNA technology entails. So basically, recombinant DNA technology involves the manipulation of DNA to create new combinations of genetic materials. This allows us to engineer and produce molecules that were once the domain of nature.
The human microbiota, consisting of trillions of microorganisms living in and on our bodies, plays a crucial role in various aspects of human development. This complex community of bacteria, viruses, fungi, and other microbes plays a vital role in our health, immune system, metabolism, and overall well being from birth throughout our lifespan. Microbiota composition analysis can be performed using rDNA technology as a form of medical diagnostics. Recombinant DNA or rDNA technology has various applications in investigating microbiota, offering powerful tools to analyze and understand the composition, diversity, and functional aspects of microbial communities. rDNA technology allows researchers to analyze and identify the microbial communities present in the samples, which are obtained from test subjects. Primarily, the microbiome composition is analyzed from bacteria-derived extracellular vesicles, or EVs, which are found in the plasma. These EVs may contain genetic material, including DNA, from the microbiota. It can then be used as a potential biomarker for designing prediction models for certain hereditary diseases. rDNA technology is used for DNA extraction from these extracellular vesicles, enabling the analysis of the genetic content of the microbiota within the EVs. This allows for taxonomic assignment to be performed, and then the microbes present in the sample can be identified and categorized accordingly. rDNA technology is utilized for pattern analysis to compare microbiome composition between different groups, such as patients with a certain disease, those with similar symptoms but are not necessarily diagnosed yet, and healthy adults. The applications for rDNA include disease prevention, biotechnology, and metagenomic studies. For disease prevention, rDNA diagnostics allows for the early detection of specific genetic markers that are associated with diseases, including but not limited to genetic predispositions and mutations linked to various conditions such as cancer, cardiovascular diseases, and neurodegenerative disorders. By analyzing an individual's genetic makeup using rDNA technology, healthcare professionals can predict susceptibility to certain diseases, enabling personalized preventive measures, lifestyle modifications, and regular screenings to minimize the risk or detect diseases at their earliest, more treatable stages. rDNA diagnostics can provide valuable information for genetic counseling, which can allow individuals to make informed decisions about important things such as family planning, potential health risks, and preventive strategies to mitigate the impact of inherited genetic conditions. In the field of biotechnology, rDNA technology is very fundamental to genetic engineering and the modification of organisms for beneficial purposes. In biotech, rDNA is used to introduce, delete, or modify specific genes in organisms, facilitating the production of pharmaceuticals, biofuels, and industrial enzymes. rDNA technology plays a critical role in the production of recombinant proteins, including therapeutic proteins such as insulin, growth factors, and vaccines. By incorporating specific genes into host organisms, scientists can produce proteins with desired characteristics for medical and industrial applications. These types of rDNA techniques are employed to create genetically modified crops with improved traits, such as resistance to pests or tolerance to environmental stress. This definitely has significant applications for agricultural biotechnology and global food security. In metagenomic studies, on the other hand, rDNA technology, particularly 16S rRNA gene sequencing, allows for the comprehensive analysis of microbial communities in various environments, including the human microbiome, soil, water, and air. Metagenomic rDNA studies contribute to the discovery of microbial biomarkers associated with specific diseases or environmental conditions. This information can be crucial for disease diagnosis, environmental monitoring, and understanding the impact of microbial communities on ecosystems. rDNA technology helps unravel the functional potential of microbial communities by analyzing the collective genetic material that is found. This includes identifying genes involved in metabolic pathways, antibiotic resistance, and other functional attributes contributing to a deeper understanding of microbial ecology and evolution.
In the realm of modern medicine, the diagnostic process in recombinant DNA technology stands as a pioneering method to detect and identify specific genetic sequences linked to diseases and conditions. This intricate process follows a systematic series of steps leveraging genetic engineering techniques and molecular biology principles. The journey begins with the collection of biological samples from individuals suspected of harboring a particular ailment. These samples, ranging from blood to tissue, contain the genetic material essential for analysis. Once obtained, the DNA is carefully extracted and isolated using specialized methods, extracting the very essence of genetic information crucial for the diagnostic process. Next, the target DNA sequences associated with the disease or condition undergo amplification. Techniques such as the polymerase chain reaction or PCR come into play, multiplying the copies of the desired DNA sequences and simplifying their detection. Precisely designed DNA probes or primers are then introduced. These probes are meticulously crafted to match and bind specifically to the targeted DNA sequences. In a process known as hybridization, these probes seek out and attach themselves to the complementary DNA sequences present in the sample, forming a crucial connection. Detection methods vary, employing fluorescence, radioactive labeling, or molecular markers that enable visualization or quantification of the bound DNA probes. These techniques facilitate the identification of the hybridized DNA, indicating the presence of the specific DNA sequence associated with the disease or condition. The obtained results are rigorously analyzed and interpreted, forming the basis of the diagnostic conclusion. Through stringent validation measures and quality control, the accuracy and reliability of the diagnostic tests are ensured. The final results, showcasing the presence or the absence of the targeted DNA sequence, are reported, a pivotal piece of information guiding healthcare professionals in disease diagnosis, prognosis, treatment, selection, and patient management.
The application of recombinant DNA or rDNA technology in diagnostics offers several advantages, contributing to more accurate, efficient, and targeted methods for detecting and identifying diseases. Some key advantages include precision and specificity. rDNA technology allows the design and production of highly specific probes and primers that can target unique genetic sequences associated with particular diseases. This enhances the precision of diagnostic tests, reducing the likelihood of false positives or false negatives. Next is early detection. By detecting specific genetic markers or nucleic acid sequences associated with diseases, rDNA-based diagnostics enable early detection of infections or genetic disorders. Early diagnosis often leads to more effective treatment and improved patient outcomes. Next is quantitative analysis. rDNA technology enables quantitative analysis, allowing for the measurement of the amount of a particular nucleic acid sequence present in a sample. This is crucial for monitoring disease progression, assessing treatment efficacy, and determining the viral or bacterial load in infections. Next is high sensitivity, the amplification of DNA or RNA sequences through techniques like polymerase chain reaction or PCR enhances the sensitivity of diagnostic tests. This is particularly beneficial when dealing with low concentrations of pathogens, providing a higher chance of detecting the presence of the target nucleic acid. Next is versatility. rDNA technology can be adapted for various diagnostic applications, including the identification of pathogens, detection of genetic mutations, and screening for hereditary conditions. This versatility makes it a valuable tool across different fields of medicine. And lastly, genetic marker identification. rDNA technology facilitates the identification and characterization of specific genetic markers associated with various diseases. This knowledge is essential for understanding disease mechanisms and developing targeted therapies.
Recombinant DNA technology has greatly advanced diagnostics, but it comes with notable drawbacks. High financial costs hinder accessibility, especially in resource-limited areas. Technical complexity, particularly in procedures like PCR and DNA sequencing, requires skilled personnel, raising the risk of errors without proper training. Sensitivity to contamination poses a risk of false positives, demanding meticulous sample preparation. Ethical concerns arise, addressing issues of genetic privacy, consent, and responsible use of sensitive information. The technology's effectiveness varies for different pathogens due to complex structures and evolving genomes. Despite challenges, rDNA has improved early disease detection and personalized medicine. Ongoing research seeks to overcome limitations and enhance the overall utility of molecular diagnostics. That's all for the discussion of diagnostics as an application of rDNA technology. We hope you learned something from this video. See you next time!